So the new platforms, the mRNA platform, is where mRNA is introduced. This is not necessarily the actual entire viral mRNA sequence.
Not all of it.
It's just a certain amount that codes for a protein. In case of the coronavirus, it's the S protein, what's so-called spike protein.
Correct.
The protein that allows the virus to attach to the mucosal cells in the body, and it allows it to enter the cell. That spike protein sequence is introduced into the body. And so once it gets into a cell of the body, that particular protein and only that protein and nothing else is produced. And so if it's only that protein that's produced of the virus, there's no other part of the virus that is in the system except for that spike protein. And so, the virus is not replicating or self-replicating. All that is happening is that you have introduced that antigen, which is the spike protein against which we're producing antibodies, in this unusual way. I mean, it's very unusual because one of the thoughts is, well, why didn't you just get a whole bunch of these spike proteins, in the lab and just inject it into your body? Would that not have worked somehow?
That's true. No, the Sanofi vaccine. The Sanofi vaccine is that, actually. So Sanofi GSK is actually formulating a vaccine that is exactly that. And the issue there is, in order to develop the same immune response, you need what's called adjuvants. So you need immune stimulators in addition to the spike protein. And HPV has this, hepatitis B also has this, is you have to find the right combination. Actually, Shingrix also, if I remember right, is also protein-based. And so you got to find the right adjuvants to add to the mix to get the immune response to just the protein. But Sanofi's absolutely, actually, Sanofi and Novavax, I think that's right.
Novavax or the antigen-based, just the actual antigen.
They're looking at antigen solutions. Yeah.
So the novel concept of this is that the antigen that causes the antibody response, the protein that causes the antibody response, is that it itself is not introduced in the body. The genetic code to make it is introduced by the body. Correct. And then the body itself makes that antigen, and then the immune system responds to that. And it's a bit complicated. I mean, if there was a diagram, I think it would be a little bit easier to explain.
Yeah, that's one of those things that's easier to draw,
you just can't make it simpler than what we just said, right? That's just how that works.
Well, I mean, and that's why I kind of maybe went through too much, but like that idea that mRNA makes your body make proteins. That's how your whole body works. Any protein in your body is made that way, from your own DNA to RNA to protein. Yeah. And then in this case of the virus's RNA is made into protein. And now in the case of the vaccine...
another way to maybe explain it is that all that DNA does in the body is that it is basically software and code for proteins. And it's a protein that does all the stuff all the bodily stuff of the cellular stuff is done by proteins. They're the workhorse. Proteins are conjugated with other things as well. So it's not just purely proteins, but all life begins with some kind of protein that is encoded by the DNA. The way that genetic code gets from the nucleus, which is where, of course, the DNA is, into the cytoplasm, which is outside of the nucleus, is through this mRNA, M standing for messenger RNA. Correct. So the messenger RNA basically just takes the code. Hey, here's the code. Here's what we're transcribing today. Right. And then that protein is made. Sure. And so in this very unusual, novel way of this mRNA vaccine, we're just sort of hijacking that middle step where we're saying, here's the mRNA you're supposed to, here's the new thing we're making today. And that new thing happens to be the spike protein. So I hope we've explained that as simple as possible.
It's one of those things, again, pictures worth 1,000 words. You just slip something in there. But and the reason I keep doing that is that in this pathway, right, what the new platform has done is taken us back a step. Rather than trying to introduce the protein, we're going to introduce the RNA that codes for the protein and force your body, your cells, to make that viral protein, which is just what the virus does. Right. But it's not the whole virus. It's just that part. So another way of saying this, to make it as simple as possible, one sentence simplicity, is that this mRNA vaccine is basically giving instructions to the body to make spike proteins. Yes, that's a great.
It is giving the genetic code to make the spike proteins and nothing but the spike proteins, so that the immune system can have an immune response against the spike protein, which then effectively is also an immune response against the coronavirus.
And that's an interesting. That last step there is an interesting point, right? And that actually, I think, is for me the biggest positive of these trials is that I think there are a lot of remaining questions. But if these trials can show efficacy, then we know from these trials and nothing else, we know from these trials that the spike protein is an appropriate target.
Right, of course.
Right?
Yeah.
So we have all these questions about safety, which I'm sure we're going to get into, right? But in terms of efficacy, that was actually not obvious. That, hey, because in fact there are other vaccines being developed against, for instance, the encapsulation, right? The capsule, right? So it was not a slam dunk,
right,
that the spike protein antibody
would have been the best target.
Be protective, right?
Yeah. So I guess to some extent there was guesswork. That's why they had, I'm sure AstraZeneca and these others probably have parallel vaccine projects going on as well, and this is the one that ended up being showing the most promise. I will say that I think a lot of the pharmaceutical companies had an instinct, quote unquote, that the spike protein was a legitimate target, because that was what was identified as the crucial protein that.
Well, that also comes out of SARS and MERS, right?
Yeah,
I mean, we had a history
and
of
coronavirus in general.
Previous
coronaviruses. We've had some experience with coronavirus.
Correct. It wasn't a de novo guess.
Yeah.
Right?
They didn't just throw a dart at the thing. Yeah. They're like, well, if we target this, we target
spike protein.
So I think it's I think that's really a settled question. I mean, and I think these vaccine trials will bear that out. That if it does decrease the efficacy dramatically, as this is
it certainly seems that way.
It seems that this is probably the right target.
The right target, and that's important, right? Because out of all the things we could target, it's important to have some idea that yes, if we keep refining. And however you do it, protein, RNA, DNA, whatever you do, right? If you come up to the idea that, yes, the spike protein is the target, that actually simplifies the field dramatically.